WES XL is a genetic test that looks for spelling changes, not only in the pieces of genes that code for proteins, called exons, but also in the closely surrounding information called introns. It also looks for small missing or extra pieces of DNA known as deletions and duplications. We also call these types of spelling changes copy number variants. If available, your provider will likely recommend that we get samples not just from the patient, but also from the patient's parents. Including parent samples may help us understand a patient's test results. If parent samples aren't available, we can still run this test for the patient alone or for the patient and just one parent. The primary reason for doing WES XL is to identify a genetic cause for an individual's symptoms. This test is phenotype driven, meaning that your provider will share the patient's clinical features with the lab to help them filter through the genetic data in an informative way. Our intention is not to report all genetic changes an individual has, but rather those that are relevant to their medical history. These changes are considered primary findings. There are 3 different results that we can get from this testing. The first is a negative or normal result. This means that we looked across all of those genes in the genome and didn't find any changes that we think are causing problems. We can also find a positive or abnormal result, which just means that we found a change in one of those genes that we think is causing part or all of a patient's symptoms. The third result is a variant of uncertain significance, or VUS. This means that we found a change in a gene, but we aren't quite sure if the spelling change causes a problem or not. In these situations, your provider might recommend additional testing or evaluations, or they might wait until we've learned more information about the change. As we learn more information, these variants of uncertain significance can be either upgraded to pathogenic or disease-causing, or downgraded to normal results. Please know that in genetics, we are learning new things all the time. So if you receive a normal result now, it is possible that we will contact you in the future with an update. One of the ways we are able to update our interpretation of gene variants over time is by connecting with other providers and researchers around the world. A tool that helps us do that is called Gene Matcher. Gene Matcher is a database that follows strict safety and privacy protocols while connecting individuals that share interest in the same gene. The goal is to help solve unsolved cases. It is up to you if you would like the patient's information submitted to GeneMatcher, if applicable. As we're looking across all of the genetic information, we can identify additional spelling changes in genes that do not explain a patient's history, but do increase the risk for certain health problems like cancer or heart disease. These are considered secondary findings. If you would like, our lab will report changes in these genes that cause medically actionable, typically adult-onset conditions. We consider a condition medically actionable if there is something we can do to minimize a risk or catch the condition early, such as taking a preventative medicine or screening at a younger age than the general population. It is up to you whether or not we report this information on the patient. If a secondary finding is identified in the patient, we do not automatically report which parent also has the variant, but can provide this information if requested, since it can have implications for the parent's health and management as well. As we've discussed, WES XL includes the genetic information found in the nuclear genome, but it can also include the mitochondrial genome. The mitochondria supply energy to our body. When they do not work properly, many body systems can be affected. It's up to you whether or not you want to include the mitochondrial information on the WES XL test report. The intention of this testing is to identify a cause for a patient's concerns. However, we can sometimes uncover unexpected or incidental information that can impact the patient and/or their family members. For example, the test may reveal that a patient may develop additional health problems in the future. If parent samples are included, it may reveal that a patient's parent has the same genetic condition as their child, that parents are related to each other, or even that one of a patient's parents is not their biological parent. It is also important to keep in mind that this test doesn't detect all possible genetic changes. So there might be more testing recommended if this comes back normal. Results of this test will be available about 10 weeks or 2.5 months after samples are received by our lab. Your GGC provider will contact you to review results and next steps. If you have any questions about this testing, reach out to your Greenwood Genetics Center providers and we'll be happy to answer your questions.